Scientists at the University of Nottingham have developed an ultrasonic imaging system which can be deployed on the tip of a hair-thin optical fiber and will be insertable into the human body to visualize cell abnormalities in 3D. The new technology produces microscopic and nanoscopic resolution images that will one day help clinicians to examine cells inhabiting hard to reach parts of the body, such as the gastrointestinal tract, and offer more effective diagnoses for diseases ranging from gastric cancer to bacterial meningitis. The high level of performance the technology delivers is currently only possible in state-of-the-art research labs with large scientific instruments, whereas this compact system has the potential to bring it into clinical settings to improve patient care. This innovation also reduces the need for conventional fluorescent labels, chemicals used to examine cell biology under a microscope, which can be harmful to human cells in large doses. The findings are being reported in a new paper entitled Phonon Imaging in 3D with a Fiber Probe, published in the Nature journal Light, Science and Applications. The researcher says that they believe its ability to measure the stiffness of a specimen, its biocompatibility and its endoscopic potential, all while accessing the nanoscale, are what set it apart. These features set the technology up for future measurements inside the body towards the ultimate goal of minimally invasive point-of-care diagnostics. Currently, at prototype stage, the non-invasive imaging tool described by the researchers as a phonon probe is capable of being inserted into a standard optical endoscope, which is a thin tube with a powerful lightened camera at the end that is navigated into the body to find, analyze and operate on cancerous lesions, among many other diseases. Combining optical and phonon technologies could be advantageous, speeding up the clinical workflow process and reducing the number of invasive test procedures for patients. Just as a physician might conduct a physical examination to feel for abnormal stiffness in tissue under the skin that could indicate tumors, the phonon probe will take this 3D mapping concept to a cellular level. By scanning the ultrasonic probe in space, it can reproduce a three-dimensional map of stiffness and spatial features of microscopic structures at and below the surface of a specimen, for example, tissue. It does this with the power to image small objects like a large-scale microscope and the contrast to differentiate objects like an ultrasonic probe. Techniques capable of measuring if a tumor cell is stiff have been realized with laboratory microscopes, but these powerful tools are cumbersome, immobile, and unadaptable to patient-facing clinical settings. Nanoscale ultrasonic technology in an endoscopic capacity is poised to make that leap. The new ultrasonic imaging system uses two lasers that emit short pulses of energy to stimulate and detect vibrations in a specimen. One of the laser pulses is absorbed by a layer of metal, a nanotransducer, fabricated on the tip of the fiber, a process which results in high-frequency phonons, that is, sound particles, getting pumped into the specimen. Then a second laser pulse collides with the sound waves, a process known as Brillouin scattering. By detecting these collided laser pulses, the shape of the traveling sound wave can be recreated and displayed visually. The detected sound wave encodes information about the stiffness of a material and even its geometry. The Nottingham team was the first to demonstrate this dual capability using pulsed lasers and optical fibers. The power of an imaging device is typically measured by the smallest object that can be seen by the system, that is, the resolution. In 2 dimensions, the phonon probe can resolve objects on the order of 1 micrometer, similar to a microscope. But in the 3rd dimension, height, it provides measurements on the scale of nanometers, which is unprecedented for a fiber-optic imaging system. In the paper, the researchers demonstrate that the technology is compatible with both a single optical fiber and 10 to 20,000 fibers of an imaging bundle, 1 mm in diameter, as used in conventional endoscopes. Consequently, superior spatial resolution and the wide fields of view could routinely be achieved by collecting stiffness and spatial information from multiple different points on a sample without needing to move the device, bringing a new class of phonon endoscopes within reach. Beyond clinical healthcare, fields such as precision manufacturing and metrology could use this high-resolution tool for surface inspections and material characterization, a complementary or replacement measurement for existing scientific instruments. Burgeoning technologies such as 3D bioprinting and tissue engineering could also use the phonon probe as an inline inspection tool by integrating it directly to the outer diameter of the print needle. Next, the team will be developing a series of biological cell and tissue imaging applications in collaboration with the Nottingham Digestive Diseases Center and the Institute of Biophysics, Imaging and Optical Science at the University of Nottingham, with the aim to create a viable clinical tool in the coming years.